Well, I think it's about time to start. So thank you very much for attending this session and welcome again to the International IVF Initiative. It's session 37, which is Reproductive Biomedicine in Space. I'm honored and excited to be co-hosting this very special session. Thanks for logging in, listening, and please ask those questions. We have a talented team who are working behind the scenes to post your questions to our speakers. I encourage you all to look at the Q&A section and avoid the chat section. The questions will be answered live or they'll be typed on the Q&A section as well. My name is Giles Palmer and it's my pleasure to introduce this subject of space biomedicine. For thousands of years, we have adapted. We have learned to change our environment to suit ourselves, and in recent times, even controlled our reproduction. But as our aspirations turn to the stars, can we really conquer this environment? I only recently discovered this quiet sector of reproductive science. But investigations have been going on for decades, and more people are working in space. Indeed, since around the year 2000, humans have been living in space constantly. So put aside the planned trip to Mars, a new focus on space tourism has become more viable over the years. But what about the reproductive hazards of space travel for men and for women? We have scientists who are looking at how space flights affect this reproductive health and investigate the effects of microgravity and the high cosmic radiation. All this work is of vital importance to the space industry, but perhaps for the future of space travel anyway, but giving also insights to more terrestrial applications too. I'll be co-moderating this session with Liesl Nel-Themaat. I think that's a Vulcan name actually. And I could not have prepared this session without her. She is the IVF lab director and assistant professor in the Department of Obstetrics and Gynecology at the University of Colorado Anschutz Medical Campus. Aside from the academic and clinical studies, the doctor, as we call her, is very active, which means noisy, on several local and national professional committees. So I hand over to you, my co-moderator, number one, engage.
Hi, Captain, reporting for duty. Hi, everyone. As always, it is an absolute honor to tune in here from Starship Enterprise. For those not familiar with our esteemed captain, Giles Palmer, he's a senior clinical embryologist skilled in lab, business, and quality management, and a very significant part of the I3 team. He's literally keeping our Starship soaring on social media. Now, it would not be appropriate to continue this session without paying homage to some of the experimental voyagers who have dared to be launched into space before us. Former fallen stars have included fruit flies, mice, hamsters, guinea pigs, cats, dogs, frogs, reptiles, fish, tortoises, monkeys, and I'm sure I'm leaving some out. Sadly, not all of those brave space cadets have lived to complete the debrief of their missions. Soar high, Albert. But those observing from the safety of their earthly afternoon tea were able to gather some valuable data on how spaceflight affects human biology. Although early results have been inconclusive at best, recent expeditions have introduced galaxies of information on the effects of spaceflight on human physiology, including reproduction. We are thus beyond excited about today's topic and have 4 very exciting talks coming up. The information therein is truly out of this world. So it is my pleasure and honor to introduce our first speaker, Dr. Virginia Wotring. Dr. Wotring received her Bachelor's of Science from Florida State University and graduated with her PhD in Physiology and Pharmacology from St. Louis University. After completing a postdoctoral fellowship, she joined NASA's Johnson Space Center as a pharmacology discipline lead. Since then, she has held many prestigious titles such as faculty appointments, science and technology integration manager, and chief translational scientist at various institutes working in space research. Last year, she moved to Strasbourg, France, to become a professor at the International Space University. Her primary field of study is the actions and effects of medications used during spaceflight missions. So without any further ado, Dr. Wotring.
Thank you so much, Liesl. I'm going to share my screen. And welcome you to space, the final frontier for physiology. I've been studying physiology in the unusual environment of spaceflight for some years now. And I'm going to tell you some things that we've learned and point out a bunch of things that we don't know yet. The very first astronauts looked pretty much the same as each other. They were not a diverse group. In fact, it's kind of hard to tell these gentlemen apart from each other. This has changed. Fortunately, we've gotten a little more diverse in our astronaut corps, and this means we're seeing more diversity in physiological responses to spaceflight too. Now, there was a reason for that lack of diversity initially. There was a decision made very early on that experience as a military jet pilot would be required for all astronauts. And there was controversy about this decision. There were a group of females who were excellent pilots and went through lots of the qualification steps to become astronauts. And this wound up in front of the United States Congress, and Congress actually weighed in on this question, and they decided that all the astronauts would be chosen from the military jet pilots. And you can explore some of this history in Cleveland, Ohio. There is a really cool little museum right across the street from the Rock and Roll Hall of Fame. If you're in Cleveland, I urge you to go see this. Now, only 600 people have been to space. And, you know, for those of us who are studying physiology and pharmacology, this is a small n right here, this 600. And that's spread over all of the time that we've been going to space. Only 14% of these people have been female. Now, in the world of good news, bad news, that was bad news. But since I've started giving talks like this about 10 years ago, we've gotten a lot better. It was 11% when I started giving these talks. So the crews that we're seeing now are including a lot more women, and that's from all of the countries who are now sending astronauts to space. Since we've been sending people to space from the very beginning, from the earliest Mercury missions, we have been studying their physiology. Initially, we didn't know what was going to happen. We didn't know if people were going to be able to eat food and swallow properly. We didn't know if people were going to be able to sleep. Every new step has been more information. And over the years, we have accrued some idea of what happens to us in space. There's lots of missing information, but we can pull together this kind of picture. We see a change in nearly every physiological system that we study. These changes are measurable. They are not always clinically relevant. They don't always cross a line into a negative or pathophysiological setting. Occasionally they do, and those generally self-correct over time. The time scale here across the axis is in months. The astronauts report a number of changes that they feel, they notice, it's uncomfortable in the first few days of a mission. At around 3 days after going to microgravity, astronauts report feeling a lot better as their vestibular system gets used to where they are, as fluid shifts that happen in the body start to settle down, the kidney adjusts fluid levels and electrolytes, the cardiovascular system responds to those fluid changes, and as do red blood cells. Some of the changes that we measure continue over time. Some kind of self-correct. Almost everything comes to a new set point. It's not where people started generally. It's what we call a space normal rather than normal. But in the case of bone metabolism and radiation effects, we expect that those will continue to accrue over time. We don't really know if these are truly linear relationships. We haven't studied them in very many people for very long periods of time. it's a big open question right now, whether that's linear or not. Every 10 years, NASA undergoes a decadal review, and this is conducted by the National Academy of Sciences. And in 2010, the review in 2010 chose to highlight gender effects. They decided that there was a critical lack of information about certain aspects of our hormonal systems and what we knew about women's physiology in space. And they recommended in the strongest terms that NASA correct this very soon. There's a series of publications that came out about this. In addition to this book, this is a book you can download for free at the National Academy of Sciences. A series of publications that came out in the Journal of Women's Health in 2014. A number of space experts, both inside NASA and outside, reviewed the data that were available already. So they looked at studies that had already been conducted. Let's go into the data. Let's examine things from the point of view of men versus women and see if there's anything there at all. And almost universally, we found that we don't have enough data to analyze that way. There weren't enough women included in the studies. There are a few differences that do hold up under statistics. We're not sure if they are actually relevant in a physiological sense. You know, for instance, there is a difference in hearing. Men who have been to space exhibit hearing deficits at younger ages than females who have been to space. Well, a lot of the men who've been to space had military experience. They were flying jets. They went through weapons training. So they may have had more environmental exposure than the women did. There are fewer women who came out of the military who are in spaceflight. So we're not sure that this is really a space difference here. We've also seen some differences in vestibular adaptation as well. And the authors of this particular article asked in their conclusions, they asked more female astronauts to volunteer for these studies. They really wanted more subjects. In the review of the musculoskeletal system, we found that the variation between particular individuals far exceeded any variability between the sexes. There's a small difference in injury rate between men and women. We don't know if that is associated with space or not. This could just be that men tend to perform somewhat riskier activities. We don't know about that. We do know that some of the exercise equipment has caused issues with people previously. Using a treadmill and microgravity, you've heard that astronauts exercise for long periods of time every day. Using a treadmill requires a harness to hold you down so that you don't just fly off the treadmill. And this harness has straps. And the women complained about this strap that was across the front. They experienced some uncomfortable chafing. They didn't like that. So the NASA engineers redesigned the straps, moved it up to the collarbone, and the women were more comfortable. The men started using this one too. They said it was more comfortable as well. It turns out the men also have nipples, and they just weren't mentioning that they were feeling chafing. As far as the cardiovascular system goes, we see occasional orthostatic intolerance in individuals coming back after a space flight. There have been episodes of syncope. We mostly avoid those now with operational measures. This happens more in women than in men, and we don't understand what the reason for that is. We have bigger concerns about space radiation. We know the radiation is there. We don't have good shielding. It's not even practical right now to think about shielding. Anything we could do to shield from radiation would be too heavy for a rocket to lift off the ground. It's just not feasible. And to put it in perspective here, the radiation on a Mars transit would be about 700 times what we get on Earth every day, ordinary cosmic radiation. Certain tissues are more susceptible to radiation, and I've shown you this particular study that is decades old. And it is the basis for a lot of decisions about who goes to space for how long. Right now, NASA only permits a 3% elevation in cancer risk. And it comes back to these kinds of studies. And you see the ovary and testes are sensitive tissues. We have concerns about that. I would like you to note here that on our overall summary chart, there is no curve looking at the reproductive system. There haven't been human studies done in this area. Liesl mentioned a number of animal studies. There have been quite a few of those. We also haven't picked apart men versus women on any of these points to anyone's satisfaction. There remains a lot of work to be done here. And we also haven't taken into consideration different practices of men and women. Just yesterday, this is, breaking news. I just made these slides. This is an excellent article in The Atlantic. You might have heard news reports about the new toilet that just got sent to station. And this new toilet has features that make it easier for women to use the station toilet and separate urine from feces. For male astronauts, that wasn't such a big deal. They've always been able to do this. And it's important because urine gets recycled into water, purified into water for drinking. Women had a harder time doing that just because of anatomy and how the toilet functions. And now this new toilet makes that easier. So the space station is now more accommodating of women, and that's awesome. Now, working with my colleague Varsha Jain, we investigated another kind of practice that was different between women and men, use of a particular pharmacological agent, and I will let her take over here.
Well, thank you very much for that great introduction with not only the physiology, but in fact the road that has happened so far to, I think, now have the space academies that now have gender equal numbers of both male and female. I think it was a long road, I believe, yeah. We just have one question before we go on. And of course, all these numbers are very small, of course, so I suppose as more and more people are working in space, we'll get more. But we have a question here from Stephen Gelatli. He's saying, are there physiological changes the same for people that go into space repeatedly? Now, I know there's been a documentary about a person who's spent a year in space, but is there more to be found with people going more times into space?
So most of the astronauts that I've spoken to say that their repeat visits are easier. It may be that their bodies have learned how to adapt to being in space. They say it doesn't take as long for them to adapt when they go back on subsequent visits. But that's a really interesting question.
I mean, I wonder if people, would look at, the second time they go up, if there's any worse effects that happen, or if they're like battled hardened in a sense. But again, as this happens more often, obviously, more and more people working in space, we'll see that, yeah.
We've got astronauts who have made, 7 or 8 trips to space, and they say it just gets easier.
Wow. And some people haven't been at all as well. So
I know, it's not fair, is it?
It's not fair, no. Thank you very much. Of course, that was the first part of the introduction and the woman's health aspect. I'd now like to introduce the next speaker. This is Dr. Varsa Jain. She's again another polymath, as all our speakers are today. She completed intercalated bachelor degree on extreme environments medicine in 2006 from the University College in London. She graduated 2 years later from the medical school from the Imperial College London, then obtained a master's in space physiology and health, again from London, but this time from King's College. She has been awarded a clinical fellowship and is currently a well-being of women clinical research training fellow. She's currently researching the endometrial phenotype in women with abnormal uterine bleeding at the University of Edinburgh this time. Along with her academic pursuits, though, she's a clinical doctor training in obstetrics and gynecology. But over the last 13 years, Varsha has paved a research path for herself with women's health in space. This has involved various placements at NASA's Johnson Space Center, and she's worked alongside leading scientists and researchers in the field of space medicine to establish an academic approach to women's health in this very niche field. So I will hand over to you, Varsha.
Thank you, Giles. So yeah, for the next 10 or 12 minutes or so, I'm going to talk to you about birth control, blood clots, and babies in space. So Virginia's given us a great introduction into how women have come into being in that space environment and the physiological changes thereof, and also just touched upon that radiation exposure too. And really just to say that that exposure is mission specific. So it depends on the duration of the mission, the actual destination, the aspects of the solar aspects at the time. And this all has an ovarian impact on cycling and on function. But again, what I'm going to highlight in this talk actually is that there are a lot of knowledge gaps. Virginia's highlighted why, but I'm just going to highlight sort of what we do know and what we don't. As we look towards the future for longer duration missions, so for example, a Mars mission, a round trip would be about 3 years. That would cover a lot of ovarian cycles, and therefore the reproductive impact, again, would be greater, we assume, but again, the levels of which would be unknown. We know at the moment that there are multiple different aspects of physiology that change when an astronaut goes into space. And really their sleep is quite disturbed, but also there's quite a lot of different stresses that they encounter. This can literally be from the impact, the G-forces of going into space, or actually the work environment that they're encountering as well. And that can lead to reproductive health impacts on the menstrual and the ovarian cycle. We don't really have human data on this, but we do know, as Liesl said before, that we've got animal studies in the mouse model. We know that you lose that cycling, you've got an effect on the estrogen receptors as well, and within the uterus. And then finally, the other thing that we also don't really know at the moment is the differences that happen in the sex steroid hormones and the gonadal function as well. We do know that if women choose to have a menstrual cycle in space, they can do so, and that's normal. But ultimately, we don't know the exact details of this. Now, men and women have had, so male and female astronauts have had children after they've been into space, with generally female astronauts having children at a little bit of a later age, and fewer children per astronaut in the women compared to the male astronauts. The other thing that we just don't know at the moment as well are the infertility rates, or if the female astronauts are getting pregnant, if there are increase in the complication rates. We have a lot more data from when astronauts were conducting space shuttle missions. So these would be short duration missions up to about 2 weeks or so. And we know that there was no increase in sort of miscarriage rates at the time, or any change in the use of artificial reproduction techniques, for example, IVF, in those astronauts compared to age match controls on Earth. But again, we just don't have the data on longer duration missions like the International Space Station missions, which are generally about 6 months, and now more recently up to 12 months. If, for example, an astronaut was going to delay her pregnancy until after she'd completed her missions, we can say that the average age of her first flight is about 38 years. So if she does that mission, then needs to come back, and as Dr. Wotring has showed, there's a number of physiological factors that need to go back to an Earth-based normal. And before she starts planning her pregnancy, we're sort of thinking that she's going to get pregnant in her early 40s. And teasing out that aging impact versus the exposure to the microgravity environment or radiation, it's just not been possible yet. So again, there's a knowledge gap there. And finally, another thing that's not known is if the female astronaut has gotten pregnant before she goes to space and she's encountered hypertensive disorders of pregnancy, for example, preeclampsia, that on Earth would have a knock-on impact on her cardiovascular risk or her risk of stroke. However, if that female astronaut then goes to space, does she require further extra monitoring? That's also not known yet. So overall, we do know some things in terms of reproductive health and reproductive outcomes, but really there are a lot of knowledge gaps there. Now on to the first part of my title, which is birth control. So we know that female astronauts have been using the combined contraceptive pill to manage their menstrual cycles since the 1980s. Now, that's quite a lot of decades of use. And originally, the dose of estradiol was something near the 100 microgram mark, which is quite high. And as the research came out and the side effects became known, that's come down slowly. And now on average, the female astronauts are using 30 to 35 micrograms of estradiol. The progestin within that drug varies. And the most common form is norethindrone, but it's just dependent on what the female astronaut feels comfortable with and what her physician prescribes. The literature states that the contraceptive, the combined pill, is definitely good in terms of suppressing ovarian function. So it reduces the risk of ovarian cysts forming in space. It reduces that risk of breakthrough bleeding. And it also helps prevent that bone loss that Dr. Wotring had just mentioned before. The female astronauts are using this in a back-to-back manner in order to suppress menstruation. And what they found is after a few months, the endometrium is so thin that you don't get that breakthrough bleeding, and it's absolutely fine. And this is possible when the female astronaut is doing her general astronaut training, if she's doing mission-specific training, or even when she gets back and she needs to recover from spaceflight, she can continue doing this. There are a number of considerations about why an astronaut might consider menstrual suppression. First one is that pregnancy is contraindicated for some pre-flight training activities like diving, fighter jet pilot training, but also obviously contraindicated in space too. But then her mission training may take her all over the world. And so compliance may be a problem with a daily pill if she's crossing a number of time zones and having to stay regular with one pill. But also, Dr. Wotring's just mentioned the space toilet. But if you can imagine the actual physical challenge of changing a menstrual product in the spaceflight environment, it would still be tricky. There are no rules and regulations about menstrual suppression in space, and it's entirely personal choice. And the female astronaut generally will discuss this with her own OBGYN. Now, when I got to work with Dr. Wotring back in 2013, 2014, I really questioned why are we using the contraceptive pill in these women when we're looking at their age group as it is, and generally on Earth, I wouldn't be prescribing an estrogen-based medication to this age group. So I really wanted to sort of evaluate, can we use long-acting reversible contraceptives like the hormonal coil or the hormonal implant? And would this suffice in terms of taking away that pregnancy risk, but also suppressing menstruation as well? Really, we looked at the whole aspect of just what we're prescribing, how this would affect the astronauts, the physiology as a whole, the advantages and disadvantages of this, specifically from an operational perspective as well. And I just wanted to provide the astronauts with more choice. So now they have that available if they feel that they want to do that. And then if they wanted to use that estrogen component of the pill, just whilst they're in space in order to benefit from those sort of ovarian suppression, the lack of breakthrough bleeding, those aspects of it, they can do whilst they're in space. Now, all of that was great, but it sort of led me on to what my current research is, which is abnormal uterine bleeding. Now, we know that AUB affects 1 in 4 women. It leads to over 800,000 referrals in the NHS every year. It's the 4th most common reason for women to present to gynae clinics in the UK. And bearing in mind, these are numbers from 2007. We don't have recent numbers, but I suspect they're a lot higher. Treatments are not specific. We're essentially just giving women a shopping list of drugs for them to choose between. They try a bit, it doesn't work, they move on to the next one. And that dissatisfaction with treatments leads to a third of women needing to resort to a hysterectomy. And AUB impacts every aspect of a woman's life. So 25% of women will experience mental health problems, which exacerbates the social isolation. It has a huge impact on their physical health. A third of women with AUB will present with iron deficiency anemia due to their periods. It has an impact on their fertility goals, so they're more likely to have miscarriages or poorer outcomes. And it has a huge socioeconomic effect. And really, AUB is affecting all of society, and it might not be life-threatening, but it is life-altering. And so I wanted to know, well, why in the clinic am I seeing women with AUB, whereas in space, women can just suppress menstruation with the contraceptive pill and it works? And we know that Vivo have classified the causes of AUB according to palm coin. So with palm on the left being the structural causes of AUB and coin on the right being the non-structural causes. So at the moment, my research that's actually come from all of that space work is, well, what's happening in women with structural causes like adenomyosis or fibroids and causing the endometrium to act differently. So they just don't respond to the pill the way the astronauts do. And they're bleeding and they're having breakthrough bleeding, or they're just bleeding horrendously. And that's all come from there. So if I can ideally try and use that precision-based approach that we're using with reproductive health research in space and bring that to women here on Earth, I feel like I'll be on to a winner. But let's get back to space. The second part of my talk is about blood clots. And I'm just going to start this off by introducing this study that was published in November last year. And essentially 11 astronauts had their blood flow of the internal jugular vein measured when they were in space. So they measured it before they went into space, about 95 days, 50 days into space, 150 days into space, and then about 40 days when they got back. Oops. There we go. Can we see that? Yeah. And so over here, there's a video that you can see that actually what came about was 7 of the astronauts either had stagnant blood flow, which is on the left, or retrograde blood flow, which is on the right. And it was found through this study that 1 astronaut actually had a complete occlusion of their internal jugular vein. Retrospectively, when they looked at the data back again, they found that another astronaut had a partial occlusion. Now, what we know from this study is that 9 of the astronauts were male and 2 of the astronauts were female. So just going back to the data that Dr. Wotring showed before, again, there's this gender difference that you can't tease out because the study numbers are so small. After that publication, there was correspondence in the New England Journal of Medicine of essentially this venous thromboembolism that had occurred in space. And that was published in January this year. It talked about how the clot was diagnosed. Within 24 hours, they used enoxaparin at the treatment dose of 1.5 mg/kg in order to treat the astronaut. And then that was reduced in order to lengthen the time that they had that treatment available in space, because you have a space pharmacy, but essentially it's not going to cover you for 6 months worth of treatment. Apixaban was then sent up and treatment was given. And then within 10 days of coming back to Earth, essentially all of those changes within the internal jugular vein were completely resolved. Now, the reason I'm talking about all of this, because this is the first ever venous thromboembolism to occur in space in the whole history of humans going into space. But when I met Dr. Wotring back in 2013, we actually were working on a project to actually understand the venous thromboembolism risk in relation to spaceflight. And back then, I really wanted to know, because this was a hypothetical situation, a thrombus had never happened in space. I wanted to know, well, what data do we have at the moment? And what data can we use in order to understand that risk? So I knew that I had, well, we've got the longitudinal surveillance of astronaut health, which they look at, they keep all of the medical and research records of astronauts who have ever been into space. And that's medical and research records before flight and after flight. So if I could get the closest data before an astronaut goes into space and the closest data from when an astronaut gets back from space, you've kind of got that intervention of space flight in the middle that could then help me tease out that space risk, because doing research in space is expensive and will take a lot of time. So this led to this, our research finally being published this year. And essentially I was looking at hematological and biochemical markers of really parameters that are related to venous thromboembolism risk that could lead to an increased risk of VTE. But what we found is you've got the normal ranges in the dotted lines, and none of the astronauts' results actually went towards a clinical significant change after flight compared to before flight. And so there really wasn't an increased risk of venous thromboembolism for female astronauts during spaceflight. We tried to tease out those astronauts that were using the contraceptive pill and those weren't. And it was easier with short duration flight because more women were choosing maybe to shift their cycles and not use the contraceptive pill in space. But we found that with the long duration missions, it was really only 1 astronaut that chose not to use the contraceptive pill. during spaceflight, and so we weren't able to tease out that pill effect, essentially. Now, a question that always gets asked is about sex in space, and whether this is happening, whether we're talking about it, whether we're going to see a pregnant astronaut in space, whether this has ever happened. So suffice to say, NASA's not going to report it, and we don't know if it's happened or not. But people always get really excited about whether a baby can happen in space, whether you can have a pregnancy in space. But I've already sort of alluded to the fact there are huge knowledge gaps there. So I think that there would be a lot that needs to be considered before we could even go down the path. And I get asked the question about whether there is a possibility of having a baby in space or pregnancy in space. And really, I just sort of wanted to put together a little table to just highlight what are the challenges, or what I think the challenges would be. So I looked at this from two different perspectives. So it's either commercial space flight, which would be short duration missions, or a longer duration mission, whether that's a 6-month mission or even a 3-year mission to Mars. There are a number of factors which I think at the moment make this, for me, not possible. I'm not saying it would never happen, but I'm just saying that there are loads of things that we need to think about. Number one, I think it would be completely unethical because of the lack of information that we have right now. It's been well documented that the psychological impacts, for example, on Neil Armstrong and Buzz Aldrin were huge when they got back from their moon mission, because people did treat them differently. So there would be an impact on that child for the rest of its life. The radiation, sleep, and stress factors we've already talked about. And if we just look at the commercial spaceflight, if you've got a woman who's paying for that spaceflight, so £250,000 or more, for 6 or 8 minutes in space, but she doesn't know she's pregnant yet. What are the risks to that pregnancy? Would she be at a higher risk of a miscarriage? We just don't know. What would be the long-term impact on that fetus if that pregnancy did survive the G-forces of launch and landing, and then being in that microgravity environment? But ultimately, if she was trying to get pregnant in the future, we have no idea how that level of radiation would have an impact on her future fertility goals. Now, if we look at long duration missions and we think about either getting pregnant in space or sending a pregnant woman into space, there is no multidisciplinary team there. Yes, we have an ultrasound available on the International Space Station, but as we get further and further away from Earth, there's more of a radio delay. And so real-time scanning with experts is just not possible. So how could those obstetric scans be performed? I've mentioned that there have been animal studies before, and in the mouse model, we've seen that organs develop in different ways when the gestation period is in space. And so monitoring those changes would be also quite difficult. How would you feed that baby? If breastfeeding doesn't work, are you going to send lots of bottles up? Every single gram of weight that's sent up into space costs a lot of money. And finally, if you're thinking about having a pregnancy in space, we don't know what the ovarian cycles will be. We know that we lose bone mass, and that could lead to pelvic fractures, for example, during delivery. And if a vaginal delivery wasn't successful, there's no way that we can do a cesarean section in space right now because of the issues with sterility and fluid dynamics. So for me, when I think about babies in space, I think of all of these things as opposed to, oh my God, this is going to be so exciting. I'm just going to leave this slide up here. Less than 2.5% of research funding within the UK actually goes towards reproductive health research. And I think we need to just start talking about women's health. I think we need to start actively promoting women's health research, because then only can we answer the knowledge gaps that we have, not only in space, but also here on Earth, and just start promoting this. Thank you.
Thank you so much, Varsha. I would like to make my contribution and answer that question for you about the sex in space. I can tell you from my experience in the Starship Enterprise that sex happens all the time in space. So I just want to clarify that for everyone. But a great presentation. Thank you so much. There is a question on the background here, and pardon me if you have answered it, but has any mammal given birth in space successfully so far?
Yeah, so they found that in the mouse model, they have had labor that's been videoed in space. The the amount of time for labor was the same as the mouse model, the mouse that was here on Earth. However, the number of contractions was doubled. And they think that's because there's an issue with the gap junctions, the gap junction protein within the myometrium. So that has happened. And
the biggest challenge, you think, with the space environment giving birth, I would assume, you know, we talked about blood clots, and I was wondering how the postpartum recovery and wound healing would be affected.
I think just every single aspect of the postpartum period would be difficult. We know that here on Earth there's a huge impact in terms of postnatal depression too. And so that, I think, is another area that needs to be highlighted, because that psychological aspect of being so far away from home would be huge as well.
Wonderful. Thank you so much again for that great presentation. I would now like to introduce our next speaker, Dr. Fathi Karouia. I hope you are proud of me for pronouncing that, Fathi. Dr. Karouia is a senior research scientist within the Astrobiology and Space Sciences Division at NASA Ames Research Center in Silicon Valley, California. He received a PhD in biology from the University of Houston, followed by no less than 3 additional master's degrees, all related in space research. His impressive resume includes lead and mission scientist position at NASA, including overseeing investigations aboard the International Space Station. His research includes the response of biological systems to space environment, the effects of space radiation on astronauts' health, and developing high throughput technology for in situ space studies. So Dr. Karouia, I hand control over to you.
Thank you. Can you hear me okay?
Yes.
Excellent. Well, thanks for the great introduction. It's my, I'm very delighted to be part of this session. So thanks again for the invitation. And so I'm here as a member of a large team, which, as you can see, there's a lot of talented people that have participated to this mission, which is Micro 11. And actually, it is also designated as the first investigation conducted on board the International Space Station where bovine and human sperm was studied. One slide in regards to disclosure. I don't have any affiliation with commercial entities or products. However, sort of, we do have Simpsons and Homer, which have been participating as our mascot for our mission. And so this is one of the patch that we have used for Micro 11. And as you can see, he's riding a sperm ball, and you see on the background the International Space Station. And actually, this is not the first time that Homer has been part of our team. Previously also, he was part of a previous mission on the shuttle. And as you can see, he has the world's fastest sperms record at 7.4 kilometers per second. So as I mentioned, I am affiliated with NASA. However, I'm not speaking on behalf of NASA, so which is very important. Secondly, I was the mission scientist for Micro 11, but Dr. Tash was the principal investigator, and so at the University of Kansas. And so again, I'm here speaking on his behalf. And then obviously I do have a lot of expertise in space biology and human spaceflight, but I'm not an expert in reproductive medicine. And so this is also very important. So the outline, I mean, obviously a lot of panelists would be talking about some of the data that have been collected throughout the years. And so I wanted to give a different viewpoint, and in particular kind of lay out how space biology project, which has been selected by NASA, is implemented. And in particular, showing with the audience some of the behind the scenes perspective. And then I will be also talking a little bit about the data and in particular in relation to the motility as well as the acromosome reaction. And then we are still currently performing some analysis. So I will be briefly giving a summary of that. And then I have a special treat at the end of the talk. So I'm not going to go into details. This has already been reviewed by the previous panelists. However, I wanted to put those slides on to give some reviews to the audience if they are interested. And so the key point here is that all the organs within the human are impacted by spaceflight. What is also important is that radiation is either additive or has a synergistic effect. And so this is also very critical. And I have added some references in relation to radiation. And then the big question is, how does the spaceflight environment, and so microgravity and radiation, impact the reproductive system? And so that led to the foundation of this particular study, which is Micro 11. And again, here the idea is that if we envision long duration spaceflight, and maybe we envision us exploring new worlds, like going back to the moon, hopefully in 4 years from now, or going to Mars, well, then comes the questions about reproduction. And so obviously, to be able to reproduce, you need to have sperm as well as eggs that are healthy and fertilized. And so in regards to the processes, obviously you guys are the expert in the area, but what is critical here, which I'm emphasizing here, is that capacitation is a critical step. And what does that mean? That means that that enables the capacity of the sperm to fertilize the eggs through a series of changes. And so those changes are in relation to motility, protein modification, metabolism, and also morphological changes. And so those are critical elements that need to occur prior to a successful fertilization. And so thanks to you, thanks to this community, now we do have means to be able to perform clinical in vitro fertilization, and that allows actually to have the right condition for capacitation. And so for the Micro 11 investigation, we'll be using those conditions to be able to assess how sperms are impacted by the space environment. So this is a snapshot of what are the many hypotheses in regards to a Micro 11 investigation. And so that was based, as I mentioned earlier, on some previous data and previous study that Dr. Tash has performed on shuttle as well as on a Russian mission. And based on the preliminary results on different model organisms, it seems that spaceflight has a negative effect on male reproductive health. And so it seems that the sperm function are impacted. And so based and building on those preliminary data, Micro 11 actually is looking at two specific aims. The first one is to determine whether the activation of human and bovine. So for this particular investigation, we're using two different types of model organisms, bovine and human, as I mentioned, and trying to assess how the motility is impacted by microgravity condition. And then the other aspect is to look at, and in particular, determine if capacitation of human and bovine sperm are inhibited in microgravity condition. And so again, those would be the two criteria for the study that was conducted on board the International Space Station. So I wanted also, as I mentioned, to kind of give you some behind the scenes images and videos and so forth. And so when we try to implement a mission on board the International Space Station, obviously, as I mentioned, a lot of people are involved. NASA is involved, but then sometimes there's also commercial partners, in which was the case for this particular mission. So Bioserve Space Technologies at the University of Colorado at Boulder was the payload implementer or payload developer. And so they have the key role in one, providing the hardware, and two, providing all the paperwork that are required and requested to be able to launch on board a SpaceX and then to be able to conduct the investigation on board the International Space Station. Here I'm giving you a kind of brief synopsis of all the different elements which are important in order to perform the operation of the investigation. And again, here there was like 13 different sets of samples by having sperm from human as well as sperm from bovine. And here are all the different kits that were packaged at Kennedy Space Center that was launched to the International Space Station. And here, obviously, you're not seeing the sperm as well as the media and the fixative. So here's a picture actually that I took during the preparation for flight. And so here we have a sperm sample which is observed by Mark. And so try to assess if the vial has been preserved and there's no issues. And then here you see Dr. Tash in the background a little bit stressed out just to make sure that we do that step and processes quickly so that the samples are not be found. And so remember that pictures, because at the end you will see a video where that pictures have been taken from. So next is a brief overview of the layout of the investigation. So as I mentioned, there were samples, sperm samples from which were bovine sperm as well as human. The bovine sperm were actually all pulled from 6 fertile bulls, whereas for the human it was 12 individuals that were used. And so those were split into 2, so capacitating media, non-capacitating media. And so you had ground-based media, flight-based media, and similarly ground-based media and flight for each condition. And so those were also the same conditions for the human sperm. And then we were taking different time points. So initially at time 0, and then we were taking images and preserving the media at different time points up to 1 hour. And certainly when we want to compare results from flight, we have to do and conduct those same operations on the ground. And so those were conducted at Kennedy Space Center, where all the samples were prepared prior to launch. And so here we have all the individuals. So Dr. Tash was responsible for the microscopy. Kevin was responsible for the activation and fixation. And then Luis was doing everything and kind of mentoring and making sure everybody was, on the timeline, because the whole idea there is to mimic all crew operations. So what does that mean? So that all operation needs to occur within 1 minute and temperature within 1 degree Celsius. So this is another view of the kind of layout on the con ops. And so again, I'm just going to go through that slide very quickly, but the idea is as follows. So we have the media as well as the sperm. Those were frozen on the station. Then prior to the start of the experiment, media is thawed. Similarly, the inoculum is thawed for a few minutes. Then the crew used the media and set it into a culture bag. And then when the sperm are thawed, then we use those two vital sperms into the mixed bag. And then we take 1 aliquot, 0.2 ml, and then we inject those into the imaging cassette. Then the imaging cassette goes to the microscope. Then we take some images for all the velocity parameters. And then we fix the samples for returning them and then doing some analysis on the ground. So that investigation actually required 2 crew members. Usually only 1 crew is needed for most of the investigation, which are implemented by space biology. But however, this operation was extremely complex, and so it required 2 crew members. So Dr. Forstels was responsible for the microscopy as well as sample storage, where Dr. Eamon Chancellor was responsible for conducting all the sample processing inside the glove box, and then conducting the activation as well as a fixation. So I do have a video. Prior to that, let me kind of give you an overview of the layout. So this is the glove box where Serena has her hands in. Then we have the microscope, which is right here. Then we have a camera, which is used. Then here you have the software that would be measuring all the velocity parameters. Then you have here, you can see the cassettes, which is used for the microscopy. Then you have a timer. Obviously, everything needs to be synchronized. Then above the crew member, actually, there's a freezer where the sample were stored. In the back here, it is the incubator where samples and the pouches to warm up them, to enable to warm up all the samples prior to activation. And then here, actually, you have a camera which enables us to visualize what the crew member is doing inside the glove box. And then here you have another camera to visualize the screen. So here I have a kind of 2 to 3 minutes video of the crew. And so the crew has a pouch there which she's warming up the samples. And so within the bag here, actually, you have 2 samples. Well, those are the human samples. And so she's going to remove one of them. And then she's going to insert it into the culture bags within which there's already the media inside. So as you can see, it's not easy crew procedures. And as you can see, everything is floating around. So that's the reason why you have a lot of Velcro there, because Velcro enables the crew to be able to fix, some of the bags or some of the tools that is required. So she just removed the lower lock and then inserted the syringe into the culture bag. And then she's mixing the sperm samples with the media, which is inside the culture bag. And so I'm not sure if you can see it, but there's a little bit of bubbles there. And so she can go back and forth, mixing the media as much as she can. So now she's removing the syringes. So again, she's strapping it because she will be using a different one afterwards. And so now she's using the second one. So as you recall, we were adding two sets of sperm, 2 inoculum into the culture bag. So this is the second one. As you can see, it's a little bit faster because more or less you saw the step which were taken to kind of add the media. So she's mixing the samples with the media. So going back and forth, making sure that this is well mixed and then homogeneous within the bag. And then what is critical there is now, that would be time 0. And so what she's going to do next is make sure that the samples are well mixed, and so she puts some pressure into the bag, making sure that there's not too much bubbles. And then she's going to take another syringe, and then she's going to take a small aliquot from the culture bag, and then she's going to transfer that into the cassette. So here, the bag here in the back is the trash where she can put all the wraps, and after those have been removed from the item, in particular here from the syringe. And so, as I mentioned, she's taking like 0.2 ml of sperm within the media. And so again, you need to try to minimize having bubbles, because obviously when you try to do microscopy, bubbles is the enemy. And then when you work in microgravity, having bubbles into the cassette could be a lot of problems to be able to visualize and to be able to collect a lot of data. So now she's putting the syringe back into the cassette, and then she's transferring the sperm inside the cassette. And then you can see the other crew is going to come in, is going to turn off the fan, because what we need to do is that there's a small window here. And so then Serena is going to transfer the cassette to the other crew member, Drew, and then Drew is going to measure the different velocity parameters. So you guys are experts in those areas, but when you're trying to assess how a sperm is healthy, and more importantly, looking at some important parameters. So what you could do, you could use a system which is a computer-assisted semen analysis system, and so that enables you to actually measure 18 different parameters of sperm motility. And so for this particular investigation, we're only focusing on 6 of them, I'm sorry. So one of them is the curvilinear velocity. The other one is the amplitude of lateral head displacement. Next one is linearity. Then there's a percentage of motility, progressive motility, and then hyperactivation. So here is a view from the camera. If you recall, there was a camera just above the screen of the computer. And so here, as you can see, actually, the recording of what we're seeing from the computer. And as you can see here, there's sperm which is moving. So this is obviously very, very exciting. And so as part of the procedures, every 10 seconds, we just change the angle of view. And so, and then we kind of still collect data. So here actually are a video which have been actually downloaded from the investigation. And so on the right-hand side is a human sperm under capacitation condition, where on the right side is also human sperm but on non-capacitating conditions. So as you can see here, sperms looks more active than on the right side, which is what is expected due to the type of media. And so then we use those images to measure the different parameters. And so here are in the situation for where we're looking at percentage of motility. Again, here it's human, and then on the right side is bovine. And so we're comparing flight condition to ground condition. So ground condition capacitating is in brown, whereas flight condition capacity is in black. So as you can see here, the ground versions kind of slightly increase, where the flight version is kind of pretty flat. So this is for the human, whereas for bovine, we can see an increase there over time, and then it's kind of flat. Whereas for the flat version, I'm sorry, this is the ground version, whereas for the flat version, this is mainly flat. And so what is critical here in regards to the motility, we didn't see any changes there that were significant, so which is a good thing, which means that the sperm were healthy. However, when we compare to the percentage of reproductivity, when we realize that here this is on the ground, as you can see on the ground, there's a sharp increase all the time, where for flight, it's kind of pretty flat. Similarly, for bovine, we can see that increase, which is expected over time, whereas for flight is pretty flat as well. And so again, here are the condition under capacitation condition. And so conclusion here is that proportional progressively of the swimming sperm is diminished in flight as compared to ground. And you can see that the difference between the two conditions. So again here, both capacitation conditions, ground condition, flight condition, ground condition, flight condition. Similarly, we were measuring also the curvilinear velocity, and so pretty flat for flight and in both conditions, but there's a slight increase in 1G condition. So when we combine all the data, and I think this table kind of summarized everything here. So when we look at all those three different parameters, and actually those three, which is progressive motility, curvilinear velocity, and hyperactivity, the expectation after 1 hour under clinical condition is that there's an increase on all those different parameters. However, for the flat condition, those parameters remain flat. And so it seems like that based on what we observe in the movement, sperm fertility is likely decreased in spaceflight. So what about all the changes besides the motility? And so here we can look at the acrosomal reaction. As you know, this is required to enable sperm to be able to become fertile. So you guys are expert in acrosomal reaction, but as I mentioned here, acrosomal is required for successful fertilization. And the way it works is that during this process, which is a very, very complex process, there's release and exposure of specific enzymes. Those are necessary for the sperm to penetrate and fuse with an egg. And so here again are some schematic of the physiology of the sperm. And here you see the head. And so those processes are critical in order for the sperm to be able to penetrate and fuse. And so what we can do, we can measure those through microscopy. And in particular, we can do some fluorescence microscopy. We can do two things. One, we can measure the fluorescence of the acrosomal membrane, which is here in red. And two, we can also measure the DNA inside the head of the sperm. So here in green is the acromosome, which is intact. Here where you can see that transition in the dye, so that particular sample is undergoing acrosomal reaction, where here, as you can see here, you don't see the green fluorescence. And so in that case, that acrosomal reaction has been complete. And here there's nothing, and so this is most likely a dead cell. So when we look at the data, and here I'm just showing the information in regards to bovine. As you know, bovine, the parameters of bovine are more significant as human, but we had similar results for human. So as you can see here on the axis, we have the acrosomal intact percentage. And the expectation is to see that decreasing over time. And so we see that curve going down for the ground. However, for flight, as you can see, is pretty flat. And so again, the expectation is to see that at the beginning of the activation, throughout this next 60 minutes, the expectation is to see the percentage of acrosomal to decrease over time. Similarly, when we look at the numbers of chromosomes that have reacted over time, what we need to see here is that we need to see the fact that all the time you should see an increase. And so And so, which is what we're observing here. However, for the flat condition is pretty flat. And so here again, the condition on the ground, see here, things are decreasing over time, which correspond to an increase in acrosomes that have reacted over time. And so again, here, it seems that under spacefied condition, it's pretty flat. And so this is obviously also, the conclusion here is that the acrosomal reaction rates of bovine sperm were normal under ground condition, Monge condition, but seems to be reduced under spaceflight condition. So conclusion here is that activation of cryopreserved human and bovine sperm in spaceflight has been achieved. And so again, this investigation was the first time, the first investigation to use human sperm in space. However, the percentage of motility did not decline, so which means that sperms were healthy. However, based on the parameters that we have measured, it seems that spaceflight impacted the progressive motility, the curvilinear velocity, as well as the hyperactivity. And so this is certainly of concerns, and those were, as I mentioned, compared to ground condition. In regards to, so again, we were able to show that the sperm were healthy, and so there was no changes in the percentage of motility. And then again, in relation to acromosal reactions, the rate of human and bovine sperm were significantly blunted in flight as opposed to ground. So again, all those elements suggest that fertilization by sperm may be significantly negatively impacted in spaceflight. So again, obviously, this is a concern. And so this was the main outcome and from Micro 11. We still have a few things to do, one of which is to do the proteomics analysis, which is ongoing. And the other one is that it seems like also there was a lot of damage from most likely exposure to radiation. The samples actually were in space for 4 months before we started the investigation. And it seems like at T0, there was a lot of sperm that were damaged. And so certainly it's concerning because again, if we envision long duration spaceflight, therefore the exposure of radiation would be higher. And so then maybe that exposure most likely will also impact the health of sperms within the crew.
I just like to thank you for that great talk. And I presume what we saw was like the first reproductive biologist in the International Space Station. I mean, that was fantastic footage. Just a very quick question: when will the results of Micro 11 be made published? I know these are very special results, and you've given us a glimpse into upcoming data.
Yeah, so currently we're working on the manuscripts. We do have a meeting that is coming up next month, which is called the American Society of Gravitational and Space Research, where we'll be showing a little bit more results. However, I envision within the next few months that publication will be out, and then you will have results in relation to all the information I've shown, as well as some other information in relation to the damage from radiation.
Great, thank you. Well, there's many more questions, but we must move on. We're going to travel back in time a little bit, and I'm just going to introduce our last, but certainly not least, speaker. But I want to introduce, it's Dr. Sheila Sanderoudin. She obtained a master's degree in clinical embryology and andrology at the Jones Institute of the Eastern Virginia Medical School. She later did a PhD in developmental biology from the University of North Texas in 2018. She is a certified high complexity laboratory director and a technical supervisor by the American Board of Bioanalysts. She has served as an inspector for the College of American Pathologists, and more poignant to this session, has served as a panelist for NASA's Space Biology Research Program. So she sees all the projects, I believe, before they actually get approved. She's currently the laboratory director for IVF laboratories in Frisco, Texas, and San Francisco in California. So it's my great pleasure to hear your talk, please, Sheila.
Thank you, Giles. Hello, everyone. It is a privilege to be on this panel with renowned experts and share my experience and knowledge in addressing this critical topic. Here's a list of my affiliations. My experience as a research review panelist for NASA's Research Opportunities in Space Biology program, coupled with my experience in human IVF, has served a strong foundation for this topic. So, as many of the presenters before me has already mentioned, why study the importance of studying microgravity and space radiation. Some of the facts include that space has been continuously occupied since November of 2000, with an average crew of 6 people aboard the International Space Station, and that travels at a speed of 5 miles per hour and completes an Earth orbit every 90 minutes. This means that these astronauts or travelers are experiencing 16 sunsets and sunrises each day. Circadian rhythms are certainly impacted by this. The Microgravity Lab aboard the International Space Station has hosted nearly 3,000 research investigations to date. As the benefits of space biology and research increase, it is crucial that the fundamental understanding of space environmental factors on reproduction are also understood to prevent adverse effects on fecundity. Just as importantly, as the duration of contemporary space missions lengthen, the deeper understanding of gender effects on reproduction is warranted to minimize risk and to ensure healthy aging and offspring of space travelers. Now, it's still unknown if the biological responses seen in experiments are a continuum or based on reaching thresholds. It is also unknown if responses require continuous or intermittent exposures, or if the sensitivity dose responses change during development. Experiments on the International Space Station often use animal models such as the zebrafish or mice to further our knowledge and understanding of these key biological aspects. This is a important slide in that it addresses reactive oxygen species. The duration of a space travel mission exposes travelers to continuous oxidative stress, even while on spacecraft. These stressors induce physiologic changes, likely mediated by oxidative damage, which has adverse effects on cellular DNA, tissues, and organs. The combination of space radiation and metabolic processes in the human body contributes to the negative effects, which include various signaling pathways. Other effects are believed to be DNA damage responses and its ability to repair transcription, gene expression, and protein regulation. What is known is that the lack of gravity, such as on the International Space Station, causes acute intergenerational changes in the quality of life. Long duration of flight cause muscle wasting, bone loss, and immune depression, to name a few. Now, focusing on reproductive biology, post-exposure to the space environment is known to disturb spermatogenesis and testicular testosterone synthesis in rodents as a mammalian model. However, spermatogonial stem cells are fairly radioresistant, allowing spermatogenesis recovery, and we'll see that in a couple of the research that I will present. Space exposures also disturbs oestrus cycling and in vitro follicular development, causing accelerated depletion of ovarian reserve and preliminary ovarian failure. There is an impact on cardiomyogenesis post-exposure, which affects embryogenesis, demonstrated by decreased heart rate and downregulated cardiomyocytes, specific genes in the developing fetus. Now, research is not limited to space, and it can be conducted in simulated microgravity environments here on the ground. To name a few of these platforms that can be used in research, partial mechanical unloading and hind limb suspension, as seen on the top left image on the screen, mimic weightlessness sensations and the microgravity environment. Microgravity simulations such as the CliniSat provides a three-dimensional chamber that continuously rotates to mimic microgravity. The top right is an image of the National Space Radiation Lab, which is in New York, which can also be utilized as part of the radio space experiments. Research is, of course, performed aboard the International Space Station, which is designed to house various experiments to advance our knowledge for systems biology. Some challenges are currently present in cross-generation studies in space, such as breeding and pregnancy loss of model organisms like mouse or the rat model. Now, microgravity at the International Space Station is a sum of reduced gravitational force and weightlessness resulting from centrifugal force of the orbiting spaceship. Mammalian embryo development is not characterized as gravity sensing, in that the embryo positioning during early division does not impact development, like some of other models like the chick embryo. There is no reported effect on fertilization. However, negative effects post-fertilization to gastrulation are well documented. Now, earlier, one of the speakers did mention that fertilization can be impacted during the space flight. However, these studies and what I'm reporting is certainly post-exposure. Some negative outcomes in early embryo development include delayed growth and morbidity post-fertilization in the mouse model. A major mechanism which contributes to these outcomes and is currently being researched is the activation of mitogen-activated protein kinase, which is a subfamily of stress-activated protein kinases. Now, looking at this from a male impact viewpoint, Matsumura et al. Reported the impact of space environment on male mice caged on the International Space Station for 35 days, then brought back to the ground. They study embryogenesis and fecundity across generations. Their research included ground control, artificial gravity group based on Earth, and microgravity group aboard the International Space Station. Sperm from the F0 population showed comparable developmental rates of fertilized oocytes demonstrated in the image A at 2 days, 3 days, and 4 days post-fertilization. Cell numbers both in the ICM and the trophectoderm were also comparable across all groups, as verified by immunostaining using cell-specific markers as seen in image B. Now, surprisingly, this is contradictory to earlier findings in which freeze-dried sperm were exposed to the space environment, brought back to ground, and used for fertilization, as opposed to the live animals that were exposed in this study. This could be suggestive that the impact of dehydration coupled with space environmental stressors were perhaps contributing factors to the differences observed. Embryo transfers were then performed with the live and the live birth rate and pup morphology were all comparable. The F1 generations were mated with their subpopulation, and the resulting F2 generation showed no significant difference. Here's another study, and this time it's looking at oogenesis, a female impact perspective. Zhang et al. Used 14-day-old mouse ovarian tissue and prenatal follicles, which were cultured under microgravity. The image on the right is a study of Pcna protein expression. Now, Pcna, or proliferating cell nuclear antigen, is an auxiliary protein of DNA polymerase. It's involved in control of DNA replication as indicators of granulosa cells and oocytes. The image shows similar expression patterns on day 0, with decrease in expression on days 2 and day 4 of microgravity exposure. The image on the bottom left shows a decrease in follicular survival, follicle survival, and structural abnormalities induced by the microgravity environment, which is a direct relationship to exposure time. In another study, now this one is a decade old, however, has cross-generational data. I wanted to go ahead and bring this to attention because there are studies following this, which then verify the findings of this study. Here, mammalian reproduction, of course, was known to be highly sensitive to environmental factors. Wakayama et al. Demonstrated this by collecting gametes and exposing them to simulated gravity via the clemostat. Oocytes were fertilized and culture was carried out under simulated conditions. Samples were collected at the zygote, two-cell, and blastocyst stages. Findings included similar fertilization rates and normality of zygotes via PN staining were performed. Similar two-cell rates were observed. as in our earlier studies that I've gone over. There were, however, a significant decrease in blastocyst developmental rate, 30% versus the 57% in the control group. There was a decrease observed in the live birth rate at 16% versus 37% in the control group. When F1 offspring were mated, similar live birth rates were reported in F2 offspring. Recent studies confirm these findings. Now, in this study, a recently published by Li et al. Explored mouse and pre-implantation embryonic development during space flight of China's SJ-10 recoverable satellite. Experiments were conducted in an automated mini incubator, which they designed themselves, and it was equipped with programmable controls for temperature, automatic micrography, and fixation of samples for cultivating mouse embryos. The study showed that pre-implantation mouse embryos can develop in space. The embryos develop through a series of cell divisions through the blastocyst stage. Here again, the rate of blastocyst formation and blastocyst quality were markedly reduced. Additionally, the cells in the embryo contain severe DNA damage, while the genome of the blastocyst developed in space is globally hypomethylated, with a unique set of differentially methylated regions. To bring it all together, here is what we know. Spermatogenesis is impacted, but function returns to normal post-exposure. Cross-generational impact is not observed. Fertilization rate is unaffected using post-exposure sperm. However, blastocyst development is decreased. Oogenesis is affected. However, no cross-generational effects are observed in the F1 and the F2 generations yet. Current studies are underway to include ground-based research on female whole organisms to microgravity and radiation, as well as gonadal exposure to study F0 cross-generational effects. Epigenetic anomalies are key factors of embryonic stress, which may impact cross-generationally. More research is needed in multi-generational studies aboard the International Space Station to bridge this gap. Cellular and molecular changes due to stressfulness of space flight and working in space may increase with prolonged exposure to the space environment. Future work to include sustained differences in buoyancy and density of embryonic stem cells adjacent to water-filled cavities such as blastocephalus or amniotic cavity should also be considered. And here's a list of my references. And with that, I would like to thank the International IVF Initiative for their tireless efforts extended to the IVF community and NASA's Space Biology Research Program for recognizing fertility as an important aspect of health for space travelers. And of course, our audience, without which we wouldn't have this at all. Thank you.
Sheila, thank you so much for that excellent presentation. I have a question here from our own Captain Palmer here, and that's related to calcium utilization, which has been shown is altered in space. So knowing the importance of calcium influxes during fertilization, it is surprising that we don't see an effect of the space environment on fertilization success, right? So I was just wondering if there's any data that maybe is indicative of downstream embryo developmental effects that could indicate that maybe our fertilization is not compromised, but the calcium influxes may not be completely normal.
Yeah, so absolutely. Much of the research that I presented, of course, is post-exposure data, meaning that sperm cells or gametes were exposed at that stage and then brought back to the ground. And the rest of the study was done on the ground, with the exception of the study that was from 2009. Now, fertilization rate is not affected, you're right. Calcium channels and influx is affected later on. However, as you notice, we see that there is an effect on the development of the embryo post-fertilization, meaning that there are key aspects of impact earlier on that are not then demonstrated until later in development. So what's more interesting than that is that the blastocysts or the embryos that do overcome those stressors and do make it to later pregnancies and then live birth, then the impact is not seen in the cross generation. So that's something certainly that is intriguing that we don't know enough about. Certainly we need to explore. Furthermore, what's key here is the epigenetic factors that we do not know enough about. So those might be more, might answer those questions in more detail. So of course we have many gaps to bridge.
Thank you so much. As you know, listening to all these presentations, it just makes you realize how little we know, and it's almost like we have to establish the field from ground zero. Although you're up in space, you're not really on ground zero, but it's just mind-boggling the things that we don't know yet. And thanks to people like you, all the people working in this, hopefully, we will keep pushing the frontier forward. I would now like to open it up to more general questions and discussion. So I'm inviting all the speakers back. And Captain Palmer, over to you.
Oh, thank you. Thank you very much for this. Fascinating presentations. We've got a lot of questions, as I said, a lot of people are with us. We must acknowledge that in the audience is, of course, Georges Ramirez, who was one of the first people that in fact had papers on reproductive biology in space, and thank you very much for joining us today. It's been so well received, this so exciting, so many things that we could speak about, but with all the things that we've said now so far, could microgravity or the lack of it, what it's like on the space, give us or reveal processes and connections with other cells which we wouldn't have seen on Earth? I mean, is there some relevance to what we're seeing in space? Sheila, perhaps, are there things that we're seeing that are relevant to what we've been seeing for so many years on the space, on Earth?
Well, we're certainly seeing that, there are certain things that we cannot quite explain in the laboratory. So of course, an embryo is one that develops freely, which is why we can develop an embryo in a culture dish for outside of the body. So it is unique in that form. So of course, we don't quite know if the environment inside of the body truly does, even looking at it from a gravitational point of view, provides a very different environment to the embryo than does the culture dish. So it's sometimes when we can't explain the outcome for an IVF cycle or the development of the embryos as related to what is seen naturally inside of the body, perhaps, yes, there could be a connection that we simply do not know of yet that microgravity or different gravitational conditions could help explain. So perhaps we do have that on Earth, certainly just, again, that we just don't know enough yet about.
Sure. One question I'd like to ask to our first speaker, Virginia. A lot of the work, of course, is in low Earth orbit, I think that was called. But what do we know about going further afield? I mean, what on earth are we doing? If we know that this is happening, at the moment at this very low altitude.
Yeah, so there's a huge difference in the environment, particularly the radiation environment, when you go to deep space as opposed to the ISS, which is only 250 miles away. We have very little information on that deep space environment's effects on people. There were a few people who went to the moon, just a few. They were all men, and it was during a time when we didn't have very good ways to monitor them physiologically. Things have really advanced since then. So yeah, this is a really huge open question, and something that NASA and ESA and the other space agencies are involved in making plans for. What are the first experiments that we need to be doing when we are sending deep space missions? And those plans are in work now. Those priorities are getting set right now.
I would like to ask the next question. This is a question that was actually asked, but I reworded it a little bit just to be politically sound. who makes better astronauts, men or women? And so I will rephrase it asking, are there differences in the post-mission recovery, both physiologically and emotionally, between men and women, and do they handle the emotional aspects equally? And you guys can all chime in on that. I predict we might have a 3-1 vote on this, but that's just my prediction. Fathi?
I mean, I think that doesn't uniquely apply to space. I think in general, women are better than men for everything in life. I mean, at the end of the day, we all come from a woman, so therefore, it's pretty obvious that women are better probably prepared for a lot of things. In regards to space, again, it's not my area of forte, but I know that from a radiation point of view, women are more susceptible to radiation exposure. So certainly there will be an issue there for long duration space flight when we envision a mission, maybe going back to the moon or going to Mars. Certainly radiation will be a big factor and a criteria, maybe one of the limiting factor, I'm not sure.
Varsha, I would like to get your input on that as well.
Yeah, so I'd actually say that men and women, because of what Virginia said, they adapt differently to the environment, but they have very different roles as well. So when you speak to the female astronauts, they will always say that they train and recruit, they train, their missions are scheduled in exactly the same way as their male colleagues. They're all up there to do a job, essentially. And so I would say they both have their place, and I don't think a single sex crew would work. I think you've got to have that balance for the people who are the best for the job. In terms of recovery, you do have a difference in the physiological factors whilst you're in space. And so therefore you will definitely have a difference in the physiology when you get back from space. I think also, just because my focus is women, you've got to think about those women that are coming back in their 40s, and then they go through, and they're coming back with bone loss, and then they're experiencing menopause where you have a heightened level of bone loss too. So you do have that aspect of it. In terms of the psychological component that you talked about, from the studies that have been done, there are no differences in the ICD-10 codes of rates of psychological conditions, essentially. There are things like PMS and stuff, PMDD, that's essentially that women might experience. But again, we don't have rates for this before they go into space or after they come back.
Okay, thank you. Captain?
Can I ask a question about the genome and, what effect, is the radiation having on this and to the chromosomes, and how long, first of all, has this been studied, but also how long does it perhaps last? Perhaps these changes, to actual DNA of the astronauts.
So this is an open area of investigation as well. There's a group at Johnson Space Center, used to be led by Frank Cucinato, who have investigated chromosomal damage in astronaut blood cells. There is a somewhat elevated rate of different kinds of breaks, single strand and double strand breaks. The repair mechanisms does a pretty good job at taking care of these. We don't know yet how much worse this might be in deep space. We would assume it would be with elevated radiation exposure, but that remains to be seen. This is one of the big open questions.
Right, thank you. And one for Fathi, and again, it's a question from the audience. DNA fragmentation of the sperm, has that been looked at, or is that going to be looked at a certain time?
So yeah, one of the side projects for this investigation was, as I mentioned at the end of my talk, the fact that indeed it seems like radiation played a role in the experiment. And the reason there is that, as I mentioned, it took roughly 60 hours of crew time, which is a lot, to be able to perform the investigation. And so we launched the samples in early April, on April 2nd, as part of SpaceX 14. However, the investigation was implemented only in late July, early August, which means that the sperm were, kept in the freezer for like roughly 4 months. And by looking at the data at time zeros, there was a slight increase of damages to the sperm and DNA-based damage. And so we speculate that this is just due to the exposure of ionizing radiation. And again, currently we are assessing that. We did some preliminary study looking at fluorescence information. And indeed, it seems like there's a higher rate of DNA damage from the samples that were in space as opposed to the one in the ground. And so currently, as I mentioned, we're kind of quantifying that aspect. But certainly it's a concern. It's a concern because here, those samples were there only for 4 months. And so as you can imagine, some of the crew members, the longest they have stayed there, they stay roughly a little bit over than 1 year. So certainly this is an area where probably NASA and other space agencies will have to focus on.
Okay, I have one last general question from the audience, and then we're going to start wrapping up. How does one, after seeing this amazing session and get all excited, how does one make the switch if anyone decides, I want to do space research and I want to become an embryologist on the mission to Mars? How do you change fields and move into the field of space research? And who do we call?
I mean, maybe I can start. Obviously, there's few programs that you can attend, and probably Virginia can speak on behalf of the International Space University. In regards to NASA, I mean, I've done a lot of outreach and educational program, and so I am familiar with all the different programs. But again, it depends of age groups, it depends of where you're coming from, it depends also what level. But roughly, there's a lot of programs of which you can attend. And that could be in internships. There's some summer programs that our center hosts every year. We do have also a program for high school students, which is called a Gene Lab. So the idea there is to enable high school students to attend and be at the Space Center and then learn more about omics in relation to flight investigations. And then if you are currently a graduate student and maybe you just graduating, NASA has a postdoctoral fellowship, which enable actually anyone. So it's not just for US citizens. Actually, I joined NASA as a NASA postdoctoral fellow. And as you can hear, I have an accent. And so I'm originally from France. And so what is nice is actually the NASA postdoctoral fellowship program, it's open to anyone. So it doesn't matter where you're coming from, as long as you have a PhD. So then the idea there is to apply to the program. It is competitive because you have to write a grant. However, then the idea there would be for you, if you get the fellowship, then to join either a laboratory within academia or within a NASA center, and then you can perform some work. For people which are more senior, obviously, many of us mentioned the space biology program. And so if we look at more on the reproduction at the molecular level and mechanistic level, a space biology program will be the way to go. Because the human research program are more emphasizing on the risk. And so this is more devoted to how to minimize long duration spaceflight from a risk perspective, where for the space biology, we're more interested in the basic science, applied science, but not as much applied as the human research program. And so on a yearly basis, the space biology program in the US, they have calls for postdoctoral fellowships, for internships, and as well for investigation. I'm pretty sure it's the same in Europe.
Would anyone else like to chime in? Okay, well, I think we are officially out of time. I would like to thank everyone for taking the time to participate in this session and discussions, particularly our 4 amazing speakers, Virginia, Varsha, Fathi, and Sheila. I truly cannot wait to see what you all do next on the space frontier. Thank you to the I3 Organizing Committee, Jacques Cohen, Peter Nage, Thomas Elliott, Sheela Sadruddin, and Mary Ann Shastet. And additional triage team members for today were Dara Berger and Anna McLaughlin. And then, of course, my co-host, Giles Palmer. It's been an absolute blast. Let's do this again. This session and the presentations will be available online soon. Then we're taking a short break for ASRM, but the next session, we will have Stranger Things. So keep an eye out for your invitation. And we really hope to see you then. Same place, same channel. Until then, live long and prosper. I'm handing you over to Giles. Captain, take us home.
Thank you, Liesl. I just want to thank everyone for watching the session today. And please do send in your suggestions on what you want to see on the i3 webinars. We do update our website and social platforms on a regular basis. So the site is ivfmeeting.com, and on Facebook you'll find us on IVF Meetings. On Instagram it's International IVF Initiative, and on Twitter, where you know we're very active, it's together at IVF Initiative. It's been a real labour of love putting together this session, which I'm sure will have a sequel. So goodbye, stay safe, and beam me up, Liesl.